Lads, this is your boy Jace and welcome to the Big Brain Busters. So in this video, I am going to be basically talking about lipid digestion and transport. So before I do that, allow me to just course you through some things that I would like you to take note of as we move through this video. So first thing I would like you to take note of are the major lipid classes in lipoproteins. So the major lipid classes are the triglycerides, which we are going to be basically talking about the most in this video, the phospholipids, cholesterol, and cholesterol esters. Then another thing that I would like you to take note of are the major groups of plasma lipoproteins. So the types of lipoproteins that are there are the chylomicrons, VLDL, which is very low density lipoprotein, LDL, which is low density lipoprotein, HDL, which is high density lipoprotein, IDL, which is intermediate density lipoprotein. Then another thing also I would like you to take note of are three lipid transport pathways. So the three lipid transport pathways in which lipids are transported in the body are the exogenous pathway, the endogenous pathway, and the reverse transport pathway. Okay, then lastly but not the least, another thing that I would like you to take note of are the types of chylomicrons. So the types of chylomicrons, of which I'm going to explain as we proceed in this video. So as you can see here with me, this is a chunk of a diagram that I've drawn so that to illustrate this. So this is just simply part of the digestive system. So we know that we eat foods that contain different kinds of nutrients, and so imagine you are from eating a lipid rich diet. So the food will pass through your digestive system, and then we know that they need to be broken down for absorption, right? So the food will run into your digestive tract, and then it will enter into the duodenum. When it enters into the duodenum, in the walls of the duodenum there you have certain kind of cells. So these cells will be activated, and then when they are activated, they produce certain types of hormones, digestive hormones known as cholecystokinin and secretin. So what does cholecystokinin do? Cholecystokinin, it goes to the gallbladder. This is the gallbladder on the liver. It goes to the gallbladder and causes contraction, and then when the gallbladder contracts, it releases its contents, which is bio, into the common bile duct. And then what happens also is that the cholecystokinin, it also contracts the pancreas, and then when the pancreas is contracted, it also releases its contents. The contents of the pancreas are the pancreatic juice, which contains things like pancreatic lipase. All right, so and also remember that food is coming from the stomach, right? So since it is coming from the stomach, it is acidic. So what we need is to neutralize that acidity. So what will happen is that secretin, the hormone secretin, will stimulate the pancreas to release bicarbonate. So when bicarbonate has been released, that bicarbonate will act to do what? To neutralize that acidity. All right, so now we have our bile salts there. So the bile salts will come into here, and then these lipids, these lipid droplets which are coming in here, they're in form of globules. So these globules are just too big for the pancreatic lipase enzyme to act on it. So the function of the bile salts is simply to emulsify the lipid globules. And then when those lipid globules have been emulsified, that pancreatic lipase, so one thing, this emulsification is one factor that is needed for the pancreatic lipase to do what? To act on these lipid droplets. Another factor that is needed is that the pancreatic lipase needs an assistant. So that assistant is known as colipase. So a colipase enzyme is also attached to the pancreatic lipase. That is another factor that is needed for the pancreatic lipase to act on the what? To act on the lipid droplets. Then last but not the least, these broken down lipid droplets, broken down lipid droplets, they are insoluble in water, and then the pancreatic lipase is soluble in water. So what happens is that there should be a lipid water interface that must be created, in which water will be dragged there in order for the pancreatic lipase to be able to act on the lipid droplets. So there will be a surface area that will be created known as lipid water interface, and this type of activation is known as interfacial activation. And then when that happens, when these three factors have been put in place, the pancreatic lipase is able to act on the what? Is able to act on these guys, is able to act on the lipid droplets. And then when the lipid droplets have been acted upon, it will break down the triglycerides, and then when the triglycerides have been broken down, they'll be broken down into what? Into fatty acids and glycerol. And then after now that happens, since they've been broken down by the pancreatic lipase, we need that guy, we need those fatty acids and glycerol to be absorbed into the cell of the intestinal tract. So for them to be absorbed, they cannot just be absorbed directly. So what happens is that the bile salts will actually come to interact those fatty acids and glycerol and form them into structures known as micelles. So micelles are simply spherical structures, okay, which are able to diffuse into the aqueous membrane, of the cells, of the intestinal cells. So this is how a micelle looks like. So this is how a micelle looks like, in which the heads are centered, and then the tails, which are hydrophilic, are on the other sides, and then the heads, which are hydrophobic, are centered here. So when that happens, these micelles now will release the fatty acids and the glycerol into the what? Into the cell. And then when that happens, we do not need the bile salts attached to the micelles. So what happens is that there is simply a proton pump here, which repels the bile salts away, and then what is entered into the cell is just the fatty acids and the glycerol. And then when the fatty acids and glycerol enter into this cell, they go to the smooth endoplasmic reticulum. This is the smooth endoplasmic reticulum. And then when they go there, what will happen is that the fatty acids and glycerol will be re-interacted, will be reacted again to form what? To form back the triglycerides. And then when that happens, they'll be transported to the Golgi bodies. And then when they have been transported to the Golgi bodies, what is the function of the Golgi bodies? It's for packaging, right? Then one thing you should know is that triglycerides are actually transported in the body in form of lipoproteins. So what is a lipoprotein? A lipoprotein is simply a lipid protein complex. So meaning that on these triglycerides, which are lipids, we need a protein surrounding it. All right, so the Golgi bodies will do what? They will coat a protein around the triglyceride, and then what will happen is that they'll put that protein. So a protein coating around the lipid is known as an apoprotein. So they will put an apoprotein there, and then to make it into what? They'll make it into a lipoprotein. So a lipoprotein contains an apoprotein and the actual lipid, and it also contains some cholesterol with it. So meaning that these Golgi bodies will put some cholesterol, apoproteins, or apoproteins also into the what? Into the triglycerides, coating the triglyceride. And then that's when this lipoprotein is able to be transported into the body properly. Okay, so the apoprotein that has been added to the lipid is known as apoB48. So an apoB48 apoprotein is actually added to the what? To the lipid complex. So what happens next after that happens? What happens next is that we need this we need this lipoprotein, which is a chylomicron. So these chylomicrons need to be transported throughout the body into the tissues. So what will happen is that they'll be deposited into the what? Into the lymph ducts. So when they have been deposited into the lymph duct, they are known as immature chylomicrons. So why are they immature chylomicrons? They need to be mature, and I will show you how they become mature. But in this case, they are immature chylomicrons, and then since they are immature, they are known as nascent chylomicrons. And these nascent chylomicrons only contain one type of apoprotein, which is apoB48. All right, cool. So when they've been deposited into the intestinal lymph ducts, So they will travel there, as you can see, through your anatomy, travel into the thoracic duct, and then the thoracic duct will deposit the chylomicrons into the bloodstream. And then when they deposit the chylomicrons into the bloodstream, the bloodstream will have to deposit the chylomicrons to the tissues, to the body tissues, right, which are the extrahepatic cells. So what will happen is that as these immature nascent chylomicrons are being transported, there is a circulating type of lipoprotein known as high density lipoprotein. So high density lipoprotein contains three types of apoproteins, which is ApoA, ApoC, and ApoE. So what will happen is that HDL will add ApoC and ApoE to the immature nascent chylomicrons. So meaning that this time this HDO is the one that is activating this immature chylomicron into a mature chylomicron by adding another type of apoprotein, which is ApoC and ApoE. Cool. So what's next? So when it has been deposited into the bloodstream, it will be transferred to the what? Into the capillary bed of the tissues. And then when it has been transferred to the capillary bed of the tissues, remember now we have a mature chylomicron which contains ApoB48, which was here, which we had, and then it also contains ApoC and ApoE, which has been added by the what? By the HDL. And then what happens next is that this mature chylomicron, it goes to bind on the endothelial lining of the what? It goes to bind on the endothelial lining of the capillaries. And then when it binds on the endothelial lining of the capillaries, they release one apoprotein, which is ApoC. So this ApoC, the function of the ApoC is simply to activate a certain type of enzyme known as lipoprotein lipase. All right. And then when these chylomicrons release an ApoC apoprotein, the ApoC apoprotein will activate lipoprotein lipase, and then this lipoprotein lipase will come to act on the triglyceride of this lipoprotein. So remember that these chylomicrons, what do they contain? They contain cholesterol, they contain triglycerides, they contain ApoE, they also contain what? They also contain ApoB48. Remember that ApoC has been released, right? So this what's this lipoprotein lipase will act on the triglycerides, and then the triglycerides will be broken down into their components, which are fatty acids and glycerol. So as they are being broken down, these tissues, which are the muscle tissues and the adipose tissue, the muscle tissues will suck out the fatty acids and glycerol and use it for what? For energy. And then what will happen in the adipose tissue? In adipose tissue, what happens is that these guys, the fatty acids and the glycerol will be used for storage, so they'll be stored in the adipose tissue. All right, cool. What's next? So now, since we have sucked out the triglycerides, right, so meaning that there will be remains. So what will remain of this lipoprotein? So what will remain is simply a whole bunch of what? Of cholesterol. So cholesterol will remain in this lipoprotein, and then since it was a mature chylomicron, and then we've released its triglycerides, so meaning that we'll remain with ApoE and ApoB48, right? So there will be a lot of cholesterol remaining in here, and then this cholesterol rich chylomicron is known as chylomicron remnant. All right, so remember now the types of chylomicrons that I told you about. We have the nascent chylomicron, we have the mature chylomicron, and the what? And the chylomicron remnants. All right, so what will these chylomicron remnants do now next? So what will happen is that these chylomicron remnants will be taken to the liver. All right, and then when they've been taken to the liver, what happens next is that the liver can either incorporate them into what? Into VLDL. Whenever you are, starving and stuff, they'll incorporate them into VLDL, and then that VLDL will provide energy to your extrahepatic cells. And then when it provides energy for the extrahepatic cells, the remnants of VLDL that remains are it will appear as IDL, another type of lipoprotein, and then that IDL, it can either be again taken back to the liver, and then when it has been taken back to the liver, or at times if it has not been taken back to the liver, what happens is that it can be converted to what? To IDL. Sorry, not IDL, but LDL. So like I said, the chylomicron remnants, after they have been what? After they have been released, they are taken back to the liver, and then when they've been taken back to the liver, the liver can incorporate them into what? Into VLDL, and then from VLDL, that VLDL can be what? Can be released and then provide energy to your extrahepatic cells, and then the remnants of VLDL, or the remains of VLDL, will appear into your system as IDL, and then when it appears into your system as IDL, that IDL can be taken back to the liver again, or it can be converted to LDL, which is low density lipoprotein, and then that low density lipoprotein, it is actually deposited to your tissues. All right, so that is why LDL is known as the bad fat. Okay, it's known as the bad fat, and then HDL is known as the good fat. So like we have said, remember also, we also talked about the three pathways, the three pathways in which lipids are transported, right? So just a summary of whatever I've said. So the three pathways, as you are eating like that, as you are eating, the pathway, as the food is passing through here, as the lipids are passing through your digestive system, this pathway is known as the endogenous pathway. Endogenous pathway in which lipids are being transported. What's next? So we have the exogenous pathway. The exogenous pathway is in which the chylomicrons are transported out of your digestive system into the what? Into the bloodstream, and then straight to your tissues. That is known as the exogenous pathway. And then when the chylomicron remnants are being taken back to the liver, this is known as the reverse transport. Reverse transport pathway. All right, so this is the reverse transport pathway. So this is the endogenous pathway through your digestive system, exogenous into the bloodstream, and then reverse transport pathway. All right, and then let's run through the type of apoproteins that are found on each lipoprotein, right? On each lipoprotein. So remember that we had the first type of chylomicron, which was the immature type, which is the nascent chylomicron. It only contains apoB48 apoprotein. All right, and then the next type of chylomicron, which is what? Which is simply the mature chylomicron. So the mature chylomicron contains what? ApoB48 and ApoC and ApoE. And then what's the next type? We have the what is the remnant? The remnants only contain what? ApoB48 and Apo E. That is what the chylomicron remnants contain. And then for VLDL contains ApoB100, then it at times contains ApoE and ApoC. Then when we come to LDL only contains ApoB100. Then when it comes to IDL only contains ApoB100 and ApoE. And then when we come to HDL, remember, like we said here, HDL, it contains ApoA, and then at times ApoC and ApoE, in that these guys are released. All right, so that's it for this video. Thank you very much for watching. Please don't forget to subscribe and share the video.